Welcome everyone, and thank you for joining us today for our webinar titled Harnessing EEG Methods to Improve Basic and Translational Research for CNS Disorders. I'm Liam Sanyo from Inside Scientific, and I'm very pleased to be your host for today's event. This session is sponsored by Data Sciences International and will feature Dr. Robert Gould and Michael Girand, who will discuss methods and applications for the use of EEG to study sleep and disorders of the central nervous system. Rob and Mike, thanks so much for being with us today.
Thank you.
Thank you.
Rob, before we get started, why don't you just tell us a bit about your research background and what kind of research initiatives you have in the works?
Sure. So I began my graduate work at Wake Forest University conducting translational studies in non-human primate models of drug abuse. And then went to Vanderbilt University and worked in the Department of Pharmacology and the Center for Neuroscience Drug Discovery. And my task there was really to help develop an EEG platform to help their drug discovery efforts there. And I'll show one example of how I utilized it there for some of the drug discovery efforts. And really, throughout my career, I've wanted to do as highly translational research and study designs as possible, and that's really where I'm starting now as an assistant professor at Wake Forest, is going back to my roots in drug abuse research, but pairing that with EEG and fMRI, and really trying to increase the validity and the translatability of classic animal models of drug abuse by using some of these biomarker strategies like EEG to help understand changes in the brain and use those to develop better treatment utilities for substance use disorders and other CNS disorders.
Fantastic. Well, can't wait for the presentation. And Mike, tell us a bit about your role at DSI.
Sure, thanks, Liam. Actually, prior to my role before DSI, my background comes from working in the pharmaceutical research space as a scientist and a study director for nearly 12 years. Working in discovery labs, toxicology, and safety pharmacology environments, I came to appreciate technologies that really helped me as the researcher make my life easier. So transitioning to DSI simply made sense. Regarding my research initiatives, although I'm not conducting research right now, my role is still research-based, and my focus at DSI is to help create a collaborative relationship between us and the scientists. And truly, it's my hope that the background and experience that I have as a former scientist helps put researchers' minds at ease, as I truly want to be able to help, basically tell the researcher that we're in this together, and we want to create a long-lasting relationship that will grow as their research continues to evolve.
Perfect. Well, without further ado, I'm going to hand controls off to you, Rob, and the floor is yours whenever you're ready.
All right, so everybody should see my screen in the beginning of the presentation here. First, I just want to say thank you to Inside Scientific and DSI for really giving me this opportunity. Hopefully what I can convey to everyone today is how excited I am to be using EEG in my studies day in and day out, and how I really think they can really help increase validity of a number of different studies and research avenues across neuroscience, behavioral pharmacology, and a number of other fields. And so with that, I'll jump right in. Really, I want to get across the point today that there's a critical need for studying sleep in many models of CNS disorders. I'll use two examples. I'll use EEG as a biomarker of CNS function and really show how this can be used to aid drug discovery processes, drug discovery programs. The example I will show is data that's published that I did collect and conduct during my time at Vanderbilt University, showing some of the studies we've done surrounding an M4, a muscarinic subtype receptor, M4 acetylcholine receptor positive allosteric modulator, and how it potentially may have new avenues and therapeutic indices for treating some of the symptoms of schizophrenia. And then I'll also use another example, which is actually not from my lab, but a really elegant study design using EEG and sleep studies to strengthen the validity of a preclinical animal model, and in this case, a rodent model of cocaine use disorder. I'll show this one because this is actually very similar to the trajectory of where my research is going to go in the future, and I'll talk a little bit more about that as well. So this is a big table, and I don't want anybody to really dwell on it too much, other than to really understand and appreciate all the different conditions that are listed here. Sleep disturbances are prevalent across almost all developmental, neuropsychiatric, and degenerative conditions. So you think of some of your classic ones: Alzheimer's, Parkinson's, schizophrenia, depression. But there's also ADHD, autism, Fragile X, post-traumatic stress disorder, alcoholism, and drug abuse that all have different aspects of sleep disturbances that are associated with these conditions. And it's not hard to really understand why. If you think about the different neurotransmitters and neurochemicals in the brain that actually contribute to any of these different disease conditions, abnormalities in acetylcholine is one type condition that you think of when you think of Alzheimer's disease. You think of alterations in dopamine function associated with schizophrenia, or serotonin, norepinephrine, a number of these other neurochemicals or neurotransmitters that are disrupted with different CNS disorders. All of these chemicals and neurotransmitters in some way or another control or contribute to either waking states, as kind of diagrammed on the left with the ascending reticular activating system, or in regulating different sleep stages as shown on the right-hand panel. So in this regard, if you have any disruption in any number of CNS neurotransmitter or neurochemical, you're going to get potentially some slight alterations between wake and different sleeping states. And these can be very pronounced in terms of the inability to actually switch from waking to sleep states, or less pronounced in terms of actually transitioning from non-REM or slow wave sleep to REM sleep, or even more subtle in terms of decreased sleep quality. But all of them play a role in a number of these regulatory conditions. Two examples that I want to highlight here is the relationship between primary symptoms and sleep. So in schizophrenia, there's actually been several associations between the degree or the duration of sleep deprivation and a direct correlation with the risk of psychotic episodes. So if you think about that right there, if you could actually normalize some of the sleep impairments, you could potentially reduce risk of psychotic breaks again, other psychosis, so psychotic episodes. The flip side of that is some antipsychotics, current antipsychotic medications, can actually exacerbate some of these sleep disturbances. So some of these medications are actually treating one symptom and exacerbating another. It's also prevalent with Alzheimer's disease. Sleep deprivation has been recently linked to increases in A-beta plaque and tau accumulation. So if you're not sleeping well, it actually can potentially contribute to the progression and development of Alzheimer's disease. One of the current treatments, one of pretty much the only type treatments for Alzheimer's disease, acetylcholinesterase inhibitors, are given to patients with Alzheimer's disease not to treat the progression and prevent degradation, but really just to treat some of the symptoms and to improve arousal and increase memory performance. And unfortunately, one of the side effects with these medications is that they can actually keep patients awake at night and disrupt some of the sleep cycles as well. So again, it's a current adverse side effect on sleep that was not previously appreciated of current medications. So there's a clear need to really consider sleep both as a symptom and a contributor to disease progression, and also consider it as a target for potential novel treatment development. So when I think of EEG, I really think of two different study types, and I'll give examples and data to show how I'm utilizing both of these in my studies. One is your typical polysomnography type study, where you're really looking at sleep-wake architecture. And what I think is really powerful about this is that the mechanisms regulating sleep are highly conserved across species. So we can really design studies that can be done in humans, in monkeys, all the way down to rats or mice. And really get very translationally relevant information from these studies. The second type is quantitative EEG, and this is really what most people think of when they think of EEG, and this is a measure of neuronal activity. And you can look at baseline neuronal activity, you can look at changes in response to a drug treatment, look at pharmacokinetic to pharmacodynamic relationships, look at brain penetration, see from a drug discovery perspective, is a pharmacological agent actually getting into the brain? Look at compound target engagement, and then also in the clinic or your animal model, use it to monitor or diagnose disease progression. So really a lot of flexibility with these type of study designs. When I talk about EEG, I just want to give a little background on how I go about doing some of these. One of the questions that was presented even at the onset of this webinar was, how do you actually do some of the surgeries and conduct these studies? So I am using several of the products from DSI. One of the transmitters that I've used in the past is the Quad ET 4-channel CNS transmitter, as shown in the top right diagram here. You see one piece of it is a battery, and then the other piece is the actual transmitter that has the biopotentials or the wire leads extending off of it. And this can really be implanted either subcutaneously or intraperitoneally into your organism. In this case, the diagram of the rat on the bottom right figure there shows that the transmitter and the battery essentially looks like a little backpack on the rat that's implanted. You then probe the wires up to the skull, and what's really nice about this is there are 8 wires. You record the differential electrical activity between 2 pairs of wires, so there are 4 channels with this device, and you can really use them for any sort of biopotential that you want. The way I've done this is looked at 3 different sets of EEG recordings to look at potential brain region selective effects. So I've looked at recordings from wires placed over the frontal cortex, the parietal cortex, and the occipital cortex in rats, and then 1 pair of electrodes are also implanted into the neck muscle to record EMG, or muscle activity, which is really pretty critical to understand and distinguish REM sleep from non-REM sleep or waking periods. When I say that I tunnel these wires up to the skull, I'm not doing deep electrode recordings. Everything I'm doing is drilling a hole into the skull, as diagrammed in the middle panel, and then just placing a wire in contact with the dura. So we're just recording overall electrical activity across the brain in this instance. You can then apply dental cement covering up the wire, suture the skin closed above the skull, and it's completely contained within the animal. What's really nice about this is it allows for a within subject assessment across a really relatively long period of time, so a longitudinal study design. This is all data that's telemetrically transmitted to a receiver beneath the home cage, so these animals are able to freely move around uninhibited with really no stress involved whatsoever. And really the limit to this is how long the battery life is associated with your respective transmitter. So this Quad ET has I believe it's a 3-month battery life, and it's not 3 months from onset to offset. You can turn these transmitters on and off with a magnet. So you could record for a 1-hour period every day for years, or do 24-hour recordings intermittently, or record for 3 months consistently. So you can really use these to however you need to design your study. Some of the considerations that I want to highlight in future slides, but also mention up front, is that the time interval for your study. Again, think about when you want to be examining sleep or quantitative EEG, whether it's during a specific behavior or across the diurnal rhythm, and when you may actually either dose with a pharmacological agent or conduct any sort of environmental or any sort of stimulus-related manipulation. One big consideration for these EEG studies is that depending on the time interval for the study, a lot of data is generated, which is both really exciting because you can really data mine a lot of different pieces of information, but it can also be a hindrance because there is a lot of data, and it does take quite a bit of time to really go through it without a really integrated and optimized data analysis pipeline. And I just mentioned in terms of flexibility, you can really place these wire leads, these biopotentials, anywhere you want to record from different brain regions. I've used very similar transmitters in both a mouse, a rat, or in a non-human primate. And what's nice about all of these is, again, because of the flexibility, in a non-human primate, you could pick based on your specific focus, whether to record from the frontal cortex, the occipital cortex, anywhere in between. And a lot of people will do this based on the international 1020 EEG lead placement in humans. So really highly translational here in the way that you can look at EEG recordings across species. So when you actually record EEG, what are we actually getting and what can we actually get out of it? So in the top left corner here, you see a raw EEG trace. The difficulty for a lot of people is, okay, that's great. Yes, that's EEG. What do you do with it? You can do a number of different parameters, different mathematical derivations of it. That the most common is a fast Fourier transformation that can be done with a number of different software capabilities. The one that I'm using here, and I'll show more of, is NeuroScore, which is a product from DSI, which is very user-friendly, and I'll show more examples of that. Following the fast Fourier transformation, you can really break an EEG trace down into different subcomponents. And so when I say different components, really it's different frequency bands or different speeds of oscillatory activity. And decades of research have really shown that these different frequencies or different frequency bands really correspond with different either different brain functions or different circuits of neuronal populations firing together. So the really slowest frequency range, this delta slow wave oscillation, is very prominent during slow wave sleep. That can actually be a direct biomarker of sleep quality. As you move down these traces, you can see the oscillations are getting much more fast, much faster in frequencies. So really your beta and your gamma frequencies here are what a lot of people think of in terms of arousal, or can correlate with different memory or cognitive task performance. I will caution you that a lot of people want to use quantitative EEG as a pharmacological fingerprint or as a signature that directly can predict some sort of behavioral or environmental state, and the difficulty there is it doesn't in the absence of that behavior. You really have to pair EEG with some sort of behavior to really correlate and know exactly what it's predictive of. After you have that association, then you can certainly look at dose-effect relationships and other manipulations and get a better idea of what that change in any one frequency or change in quantitative EEG across the board might be doing to that behavior. But you do still really ideally need some sort of behavior to correspond and correlate with a QEEG study as well. As I mentioned, you take the unfiltered EEG, you can do a fast Fourier transformation and break it down. You can then pull out and quantify the relative or absolute percentage in terms of electrical output or voltage squared. Here I'm actually looking at relative power. So this is the relative contribution that any one frequency contributes to the overall total EEG signature that was recorded. And these three figures show a spectral profile or a periodogram across the different frequencies. So on the x-axis, you're looking at frequency from 0 to 100 hertz. So that's going from your very slow frequencies to your very fast frequencies, and looking at the different profile in your different arousal states. So in a waking state versus non-REM or slow wave sleep versus REM sleep. And what's really critical to point out here is to the naked eye, it may not look that different, but each one of these states, wake, non-REM, and REM, has a slightly different spectral profile. So if you were looking at what an environmental stimulus or a pharmacological agent might do to quantitative EEG, you'd really want to make sure that you're looking at it specifically in one of these states so that you're not averaging everything together and kind of washing out or increasing variability associated with your experimental manipulation. And really it's these different periodogram profiles that you can use to then identify different sleep states, whether your organism is in a wake, a non-REM, or a REM state, if you will. And so we can really pull out a lot of information that way. Another question that was presented prior to the start of the webinar was, how do you actually determine sleep states? And I'm not going to go into a huge amount of detail. I'm happy to help talk with individuals at a later time offline about exactly how I do it. But in general, there are characteristic oscillatory patterns that are associated with waking states, non-REM sleep, or REM sleep. And you can either go in individually in 5 or 10 second epochs and identify those states manually and say this specific state is REM sleep, for example, or you can use an automated sleep scoring software which really pulls out patterns and ratios of the different frequency bands to identify the different states. So here's pretty much how I do it. This is data that's opened in NeuroScore software. This is looking at a 1 hour window and just the EEG trace. And really the only thing I want to point out here is that you do see these different patterns, so different peaks in amplitude that then go down, and these would correspond with different states. We can also pull up other parameters that are recorded as well. So this is EMG and activity. So anytime you're getting a measure of activity, you know that the animal is moving around. As you can see, a number of those activity spikes also correlate with increases in EMG. Again, this is muscle activity. So we can pull out when an animal is awake and whether they're actively engaged in something and running around or eating or moving versus something where they're more passively awake or quiet awake. From there, if the animal is not awake, if they're not actively moving, if there's not a large amplitude increase in EMG, you could generally think of the animal either in a quiet waking state or sleep, and that's when you really move to the EEG trace to really pick out that specific pattern. And all of this is a big scale, so this is a 1 hour window. We can zoom in to really look at those different states. In addition, this is the spectral profile where you can look at the different distributions across EEG stages. So these different colors here represent different frequencies. So the red is delta power, blue here is theta power, green is alpha, orange is beta, and black is gamma. And you can see this different relative ratio from one pattern to another. If we zoom in, so here I'm just going to zoom in to a 5-minute window that's highlighted here in yellow. You can see more definitively EMG is very low. You're getting higher amplitude EEG. So this is slow wave, which is associated here with higher relative amounts of delta power. And then you can see a nice transition into less amplitude frequency here, which has a higher prevalence of theta power. So this is actually showing that transition between slow wave sleep into REM sleep. This is still a 5-minute window. You can zoom in even further and really look at that transition, really see more of those slow waves in a 1-minute window here. But depending on the frequency of your recordings, you could really zoom in to a millisecond level, and you could really look at event-related potential changes as well on the millisecond subscale. So with that said, after you've actually determined different stages based on their representative EEG traces as shown on the right, you can pull out a sleep statistics report from NeuroScore and then graph your data in any sort of bins that you'd like. So on the left here, I'm looking at 24 hours. So the time is on the x-axis. The first 12 hours are when the animals were in the light cycle, and then you see right in the middle when the gray rectangle pops in, that's when the lights go off, and they're in the darkness for 12 hours. And as you see in the top figure, animals are awake more when the lights go off. They're more active in the dark. They're nocturnal, whereas they sleep more, shown in the middle panel with the amount of non-REM and then REM on the bottom, more prevalent during the light phase. I will point out that even though they have higher prevalency of sleep when the lights are on, rodents actually cycle in and out of all 3 states, wake, non-REM, and REM, almost every 8 minutes. So there still is waking periods, even in the light period here, about 20% of the time. With this said, just like classic diurnal rhythms in sleep and waking states, there are diurnal patterns in the spectral profile as well. So this is just an example showing that delta power in red, alpha power in green or that high gamma frequency from 50 to 100 hertz in blue does fluctuate across the diurnal rhythm. And this is really important because this to me shows really the dynamic range across a 24-hour period. I've had a number of discussions with people that say, you know, I've done a cognition study, and in a novel object recognition task, I get sniffing of the novel object 6 more seconds, and that's statistically significant. Well, 6 seconds sniffing a novel object may be statistically significant, but is it really physiologically relevant? How does that translate forward? And what I like to really point out here is that even seeing these fluctuations in power bands or this quantitative EEG analysis, you can do a comparison between any point, any time of day here. So I'm going to normalize the time 2 hours into the dark period and look at it as a % change from 2 hours into the light period. So I'm going to take power across the entire spectral band at that time point that just popped up in yellow, normalize everything at that point to 0, such that anything that is now shown as a change from this 0 represents either an increase or a decrease in power at a different time of day. And so if we look at this, look at what's happening 2 hours into the dark period compared to 2 hours into the light period, what you see is about a 100% increase in high frequency gamma power across the diurnal rhythm, just from the active period in the dark as compared to the light period. This gives me that dynamic range. It really says a physiologically relevant change across a normal, healthy rat. You can look at about a 50 to 100% change across time. So really with any sort of pharmacological manipulation, we wouldn't want to be much more above that, or we wouldn't want to be above that per se. You could potentially incorporate that in a number of different ways. You could either be looking to increase or decrease by 50 to 100% as a therapeutically relevant profile, but anything outside of that range might in fact be detrimental or more disruptive. And so a lot of this comes down to time of dosing. What are you looking at? What question are you wanting to ask? And where might you want to actually record in terms of your study design? So for example, if you're dosing in the light period when they're typically sleeping and they're not awake as frequently, you could be looking at either wake promoting effects if you increased % of time there, or potentially sleep disrupting effects. And it could be interpreted either way, and you really need to do the analysis to look and see how the manipulation might be affecting quantitative EEG to see if it's affecting arousal in a positive or a detrimental way. Conversely, if you dosed in the dark period when they typically are awake, you could look at either sleep promoting effects or sedative effects. And of course, this is looking over a 24-hour period. If you're looking at the effects on a specific behavior, you could certainly design a study where they're awake the entire time and it's a much shorter signal window or recording period. Along with that, you can look at the spectral profile. So if we normalized everything to that 2-hour period during the light cycle, if you decrease lower frequency power, as shown here on the left-hand part of this periodogram or change in periodogram, and increase in high frequency power, if you're doing this profile when they're in a state of relatively low arousal, it can be conceived to be potentially cognitive enhancing effects. And a number of cognitive enhancing compounds will show this profile. In contrast, if you did that same profile and were increasing high frequency gamma power above what is physiologically relevant, it could be disruptive. In fact, patients with schizophrenia actually have aberrant or heightened gamma power compared to normal patients. So this could actually be indicative of potentially disrupting or increasing psychotic-like behaviors. In contrast, if you dosed and saw the opposite profile as shown here in the red line, really where power distribution is being shifted from that higher frequency, where that's decreasing and it's shifting more towards the lower frequency, you could interpret this as potentially sedative or a decrease in arousal state. And again, really determining what this does or what this means behaviorally, you really need a behavioral or a cognitive endpoint, some sort of behavioral manipulation to go with it. You can't just arbitrarily say it's an increase or a decrease, which might be therapeutic or detrimental per se. Again, this is where EEG can really complement some of those other behavioral models. So when I talk about manipulations and other behavioral models that you could actually use for this, a number of people have just used sleep and EEG in freely moving animals to look at differences between a knockout or a knockdown or an overexpression type strain. But a number of people are now using it paired with chemogenetics or optogenetic stimulation to really look at specific circuit or neuronal activity effects on sleep or arousal. Of course, this can really be used to impact and help inform and improve validity of a number of different models of diseases. So for PTSD, Alzheimer's, schizophrenia, addiction, a number of models. You can look at environmental and social variables and how that might affect sleep or arousal. And then, as I'll show you in the next few slides, really look at a pharmacological manipulation and see how you might be able to shift arousal or sleep states and use that to inform future drug discovery efforts. So as I mentioned, I did some work towards a potential new treatment for schizophrenia. For those that are not as familiar with schizophrenia, there are three primary symptom clusters: positive symptoms, the paranoia, the hallucinations, and the delusions, which are what most current antipsychotics treat. There are negative symptoms and cognitive deficits that are also associated with schizophrenia that are largely untreated. But really the fourth type symptom that most patients complain about are sleep impairments, sleep disturbances. And most patients with schizophrenia say they have difficulty falling asleep, but when they do actually fall asleep, they drift through the stages of slow wave sleep faster and drop into REM sleep much more rapidly, which can have some serious consequences in terms of memory consolidation and a number of other physiologically relevant factors that can be disruptive overall. So really these sleep disturbances do occur in unmedicated and medicated patients, have been tied to a prodrome for psychotic symptoms, and really represent an underappreciated symptom that could be treated. We could really develop treatments specifically for this, or at least consider this when designing treatments for the other symptoms. And that's what we wanted to look at. We wanted to develop M4 selective positive allosteric modulators for the acetylcholine neurotransmitter system, and benchmark that with what Clozapine does to sleep and arousal. So Clozapine is an atypical antipsychotic. It's one of the primary treatments right now for schizophrenia, but it also has some adverse side effects associated with it, one of which is sedation. So you give patients with schizophrenia Clozapine, it will sedate them, it'll keep them from hurting themselves and others, but it certainly does not help them integrate back into society or even be able to function on their own very well. So we wanted to look at the effects of Clozapine. We're dosing here either 2 hours into the light cycle as shown on the left figure, or 2 hours into the dark cycle as shown on the right figure. We looked at a dose-dependent effect, doses that are known to have antipsychotic-like activity in other animal models of schizophrenia. And we saw a dose-dependent, really an interesting profile on time awake when dosed 2 hours into the light cycle. A moderate dose increased percent time awake initially, but then actually decreased time awake several hours, 12 hours after dosing almost here, which was an interesting profile. It had no effect on time awake when dosed in the dark active period. But what was really interesting is if you just look at this time point that I'm highlighting here, 2 hours after dosing, and pull out quantitative EEG and really looking at the pattern here, if you just focus on green, this is the highest dose, 10 milligrams per kilogram, regardless of time of dosing, it's shifting this relative power away from higher frequency more towards increased prevalence of lower frequencies. So it really can be indicative of EEG slowing, which has been shown to be more of a sedative profile in patients with Clozapine. And we know that in animals, Clozapine can actually disrupt learning and memory tasks as well. So here it's actually showing that Clozapine can show disruptive effects in quantitative EEG here. In contrast, if we looked at our M4 selective positive allosteric modulator, again, dosed, looking at time awake here, dosed either 2 hours into the light cycle or 2 hours into the dark cycle, no effect on overall time awake. But if you look at that same period, 2 hours after dosing, you're getting a subtle increase, but statistically significant increase over here in that higher frequency gamma range, which could be indicative of increased arousal or cognitive enhancing effects. And in fact, that is exactly what we were able to correlate it with. In separate subsets of animals, this compound at this dose range could actually enhance learning and memory performance. So we look at this and interpreted this as a potentially therapeutic type effect. In contrast, if you look at dosing into the dark period when they're already at a heightened level of arousal, if you will, there was no effect at all. So this mechanism really had a state-dependent effect, completely unique from current antipsychotics. So we're really excited about that. And I just want that to really highlight kind of the utility of the EEG for these type studies. The other point that was actually really interesting is if you look at this same compound, this M4 positive allosteric modulator, on time of sleep across the 24-hour cycle, it had absolutely no effect on duration of non-REM sleep, either in 1 hour bends as shown on the left panels, or in the 4 hours cumulatively just after dosing. However, if you look in the bottom panels, it had a nice dose-dependent decrease on duration of REM sleep. As I mentioned, patients with schizophrenia have difficulty falling asleep, but when they do go to sleep, they fall into REM sleep and have too much REM sleep. So this profile could not only be increasing arousal during the day when they are awake, it could also be shifting the balance between REM back to non-REM sleep. So there are several different potential therapeutic applications here. Of course, all of this was in healthy rodents, just characterizing this mechanism. So it needs to go in and be validated in a number of other animal models. The other way we can do this is one typical animal model for schizophrenia is the two hypotheses for schizophrenia are hyperdopaminergic mechanisms regulating or contributing to the psychotic type symptoms in schizophrenia. So essentially too much excessive dopamine in mesolimbic regions. So you can model that in animals by giving them a dopamine releaser like amphetamine. The other hypothesis or underlying theory is that there's decreased glutamatergic function, so hypoglutamatergic function, and you can model that in animals by giving an NMDA receptor antagonist like MK801 as shown here on the left. And essentially in both cases you increase dopamine or decrease glutamatergic function, and you actually get excessive hyperactivity in rodents. So of course translating this to a human endpoint is a little more difficult. But using EEG, you could really see what these compounds are doing in the brain and if you're able to reverse it. So here, this M4-PAM VU467154, when given prior to MK801, dose-dependently decreased hyperactivity, which is great. It's really informative in a high-throughput screen, but it doesn't tell us a whole lot about what's going on in the brain. Here, we can use quantitative EEG, just looking at high-frequency gamma power, with the same paradigm, giving either vehicle or our M4-PAM, followed by MK801. MK801 in red increases high-frequency gamma power, which could mimic some of those symptoms in schizophrenia, where you do see excessive gamma power in some patients. And our M4-PAM at a top dose from the previous figures does subtly, but does reduce the effects of MK801. In contrast, Clozapine in green showed a decrease at this top dose at baseline and below baseline even after MK801. So here we're utilizing quantitative EEG to really complement historic and classic models of antipsychotic-like activity to help us move forward and see if there might be more therapeutic and or less adverse effects compared to classic antipsychotic medications. So in this example, hopefully I got the point across of using EEG as a biomarker for functional target engagement. We're giving a compound and able to see a dose-dependent effect as it's crossing the blood-brain barrier. We don't know exactly where it's binding per se from EEG analysis, but we are getting this dose-dependent relationship. We're able to distinguish potential therapeutic versus adverse effects, both on sleep and arousal using quantitative EEG. And again, this type application, you could give MK801 or an amphetamine challenge type study design in humans. You could really use a very complementary study design in human studies in early clinical trials to see if your compound is going to have the same effect in humans that it had in animal models. With that, I'm going to shift gears and talk about using EEG to increase the validity and then translatability of your animal models. I'm really interested in substance use disorder research now. For those of you that aren't familiar, I'm sure most of you actually do know somebody that's struggled with substance use disorder, whether it be alcohol, opioid. The opioid epidemic now is huge, or others. But what's really striking is that the cost of the U.S. Is actually larger than any other brain disorder at last estimates. And with the increasing number of Alzheimer's, this may change in the next decade or two, but as of now, it's incredibly costly, more so than any other brain condition that we study and treat right now. There's a cycle of drug abuse, followed by abstinence, followed by relapse. We can model all of these in animals and try to incorporate EEG to really help enhance and understand what's going on during these different phases. We have really good animal models of drug abuse, of substance abuse. Unfortunately, they really haven't contributed to a high degree of successful treatments yet. And a lot of that may have to do with not fully encompassing some of the other behavioral symptoms that are seen, such as sleep disturbances or cognitive impairments. And that's really where my research is going now, is to focus on some of these substance-induced disorders, including sleep impairments, and try to normalize those as a way to potentially not solve addiction, but help people reintegrate back into society and be able to function more normally. If you think about how disrupted a single night of sleep disruption is for you, whether it's staying up late, watching a sporting event, a sick child, etc., you don't function as well the next day. Doing this day in and day out for months on end is very detrimental to overall daytime performance. Very quickly, I just want to highlight that a recent review showed that nearly 70% of patients admitted for detox, regardless of drug abuse, reported sleep problems. 80% of those attributed the sleep problems to substance abuse. So there's really this vicious cycle. Drug use leads to sleep disruptions, which then leads to further drug use. Another review recently came out showing that, not surprisingly, drug effects on current users are pretty class specific. Stimulants can promote waking and delay sleep, whereas depressants can promote sleep. But in general, all decreased total sleep time or sleep efficiency and quality. But there are some subtle differences between drug effects on sleep during abstinence, and these last for several weeks to months into abstinence, suggesting that there really are different disruptions following different drugs of abuse, which implicate differences in underlying neurobiological changes, and we should really understand those and try to treat those differently. The third bullet, the third main point from this review, was that actually we haven't studied these type substance use paradigms in animals, and we need to in order to fully understand what's going on and develop potential treatments. There's also an association between sleep disturbances in abstinence following cocaine use and cognitive impairments. So again, if we can think about improving sleep, maybe we could improve cognition, we could help patients hold down a job and juggle work-life balance better. As I mentioned, we have very good animal models of this. The classic animal model for IV drug use, and specifically cocaine use disorder, is to implant a jugular catheter into the jugular vein of rodents, then connect them to an infusion pump where rodents go into an operant chamber and then they lever press. And they, following a set number of lever presses, it delivers an infusion of the drug or whatever's in the syringe. What's really nice about this is, just like any of us, individual rodents do have individual differences in both sensitivity to drugs of abuse and temporal precision. So they're lever pressing, they're taking drugs at the time they want. They're actually administering it, self-administering these drugs of abuse across a 2 or 6 hour window when they want to. It's not like we're forcing it on them. In this regard, we can model all the different aspects of drug abuse, from initial sensitivity, sampling, to acquisition, escalated intake. We can then put them in periods of abstinence and see what's going on in other aspects of behavior. We can then put them back in the same chamber and show them light cues that were previously paired with drug delivery to look at how much they'll actually respond for the previously associated light cue. And in this regard, we can look at drug seeking or a model of craving or relapse, if you will. So I want to point out one more quick example. I know we're running short on time. This was not a study I did, but it's a really elegant design. So I want to highlight these studies real quickly. This was a study by a group looking at rodents that self-administered cocaine and were implanted with EEG to look at sleep durations, both after initial self-administration, but then during periods of withdrawal or abstinence. And so they looked up to 21 days of abstinence and showed that non-REM sleep actually decreased over the duration of extended abstinence period, or withdrawal as they called it. REM sleep was the same thing. It actually got worse over the course of almost a month of withdrawal. That's really powerful. This is in the absence of the drug of abuse. In the absence of cocaine, these sleep disruptions are getting worse. The duration, the episode duration, so not only can we measure total duration of these states, but actually within it, how long each REM cycle went actually decreased as well across this withdrawal period. So this reiterates that we're in the absence, during abstinence, in the absence of the drug of abuse, we're getting these long-lasting sleep disturbances. So this same study by the same group wanted to see if they could consolidate sleep and actually improve or if they could decrease drug-related behaviors by improving sleep. And so the way they went about doing this was using a sleep restriction paradigm using a treadmill during the dark period when they should be awake. So the thinking here was if they kept them awake when they're already awake for the majority of the time, maybe they could then consolidate sleep during the light period when they should be sleeping. And that's exactly what they saw. They were able to increase the amount of non-REM sleep following activity on a treadmill. They were also able to increase the amount of REM sleep, which was significant here, following just a very short duration of time on the treadmill. They wanted to show that this wasn't just a random phenomenon. They did a really nice characterization. They looked at withdrawal out to 42 days following self-administration, and in different groups, they looked at the number of days of treadmill running or sleep restriction. And what they then looked at was comparing this to that final measure when they put animals back in the chamber and wanted to look at the amount of responding to the light cue as a measure of drug seeking or craving. And what they saw here is as a result of this increased amount of sleep restriction, they were actually able to decrease the amount of responses, the active nose poke responses here, following 45 days of abstinence in a really time-dependent manner based on how much sleep restriction and then consolidation they were able to achieve. Well, I'm not going to highlight it. What they also did with this, they did the opposite. If you actually fragmented sleep further, you made this amount of behavior, this drug-seeking behavior, increase. Equally important, they also looked at markers of synaptic plasticity in the brain and showed that not only were they were normalizing sleep, they were changing behavior, but they were altering neurochemistry and neurobiology as well in a way that was predictive of a potential treatment. So it really established both a brain behavior and neurobiological association between this potential manipulation on sleep, which to me is incredibly powerful and reiterates the need for developing treatments to improve sleep to aid treatments for substance use disorder. And that's really where I'm going with some of my research now, is to look at new pharmacological mechanisms and see if we can normalize sleep and normalize cognitive impairments following substance use or self-administration in animal models. So hopefully from these studies I just showed you, I've highlighted the need for studying sleep in models of CNS disorders to really be able to establish brain behavior relationships, to use EEG as a biomarker of CNS function, to really strengthen and increase translatability of your animal models, and that the application can really be flexible and user-friendly. Very briefly, I want to acknowledge a number of people. All of the work at Vanderbilt was conducted in Carrie Jones's lab. It has previously been published, and working with the Center for Neuroscience Drug Discovery at Vanderbilt, as well as funding for a number of these studies over the years as well.
All right. Well, Rob, thanks so much for the really fantastic presentation. And moving along, we have Mike Girand, who will discuss complementary endpoints for neuroscience research. So I'm just going to pass off controls now.
Thanks, Liam, and thank you, Dr. Gould, for an excellent presentation. With respect to time, I just want to spend a few minutes today to talk about the potential and the rationale behind adding complementary endpoints to CNS-based research. And for me, it really boils down to creating some best practices, not only for your specific area of research, but best practices that the scientific community can leverage. These include an increasing demand in responsible animal use, the potential for a more robust study design, and the translational impact in both human and animal markets. And then translational impact is really the key here. Here at DSI, we have the privilege of collaborating with some great scientists, and we recognize that funding is becoming more and more difficult. However, our experience has showed us that when research has the potential for a larger translational impact, the probability of funding actually increases. In animal research, it should really come as no surprise that there continues to be a push in getting more from an animal model. Both external and internal groups stress that if we're going to use animals in research, we better be doing it the right way, and we better be taking certain things into consideration. Specifically, if in vivo research is to continue, we have an obligation to focus on the reduction and refinement of animal models being studied. That means that there's an increasing trend to do more with less, use the latest technologies, and capture a holistic view of the disease model beyond the more obvious organ system being studied, suggesting that we may need to go beyond the brain and consider additional endpoints. As study directors, we understand that there are basic requirements when designing an experiment. Some of these include clearly stated objectives, proper sample size, and using an appropriate statistical test. But an important part of a well-designed experiment is its range of applicability. For example, can the results be generalized across strains, environments, or techniques? More specifically, can the range of applicability go beyond the study of the disease model itself? Can we actually help draw better conclusions or suggest further evaluation to truly add to a scientific knowledge base? And adding additional endpoints may help us. As this slide suggests, it's really more about EEG data collection. As Dr. Gould pointed out in his talk, sleep disruptions are observed across CNS disorders. In similar fashion, no matter the neuroscience application, there is likely the potential effect or impact on other organ systems. At DSI, we work with many scientists who enhance their research by combining applications and collecting multiple endpoints. In the coming slides, I'll provide a few examples, keeping in mind that although EEG data collection is at the forefront, it's only part of the overall picture. Affecting nearly 50 million people worldwide, epilepsy is one of the most common neurological diseases. Neurocardiac dysfunction is observed, whereby changes in morphology and cardiac arrhythmias occur. Increases in blood pressure are also seen during epilepsy-type events. From a respiratory perspective, prolonged cerebral hypoxic conditions have been reported as a potential risk, and changes in core body temperature often correlate to change in stress levels and its association with seizure risk. Today, anti-seizure medications come with many and serious side effects, including or impacting other organ systems. Alternative approaches, including electrical and optogenetic stimulation, may provide fewer side effects. Nonetheless, it's important to properly evaluate its impact downstream. In the research community, mood disorders are particularly difficult to study. However, there are some objectively measurable symptoms which may be able to provide some clarity. These include autonomic dysfunction and heart rate variability using ECG or blood pressure signals, changes in frequency component during sleep, and hyperregulation of the thermoregulatory center. Some actually take it a step further by combining these endpoints with restraint and fear conditioning equipment, helping us to confirm and evaluate stress models in real time. With regard to addiction and drug abuse, the physiological and psychological effects are significant. Observations include changes in heart rate and blood pressure. More severe complications include arrhythmia and the potential for a heart attack. Increases in body temperature, muscle twitches, and spasms are sometimes observed. And depending on the type of addiction, restlessness, anxiety, and panic may take place. In the field of TBI, a few recent publications have raised questions about animal models and their ability to translate to human therapy. Recommendations from these authors suggested a consistency approach when using biomarkers. Combining intracranial pressure monitoring with other neurological endpoints can help us understand the pathophysiology of brain damage. Sleep disturbances occur in a high percentage of individuals following injury. There's an increased risk of seizure in patients with TBI. Monitoring core body temperature allows us to better understand recovery and treatment approaches. And patients with TBI often experience pulmonary complications, including acute lung injury and acute respiratory distress syndrome, whereby researchers may consider classic resistance and compliance testing. EEG is a common tool when studying Alzheimer's disease, looking at an increase in delta and theta activity. And given that there is no cure, we see a focus on identifying and alleviating symptoms. Changes in sleep architecture occur, showing a reduced percentage of slow wave sleep, broken sleep periods, and reduced REM sleep. There's often a blurred distinction between the disease and the patient's actual cause of death. Shortness of breath, pneumonia, and other respiratory disorders are observed, leading to a lower quality of life. Current research suggests a bidirectional correlation between elevated blood sugar and Alzheimer's disease. There are disturbances within the circadian rhythm, including increased evening activity, alterations in core body temperature, as well as changes in fasting and feeding cycles. And finally, recent studies identify differences in hypertensive and non-hypertensive patients with respect to cognitive test results. To summarize briefly, preclinical in vivo studies continue to evolve by maximizing the number of endpoints and biomarkers, helping to better characterize the model being studied. This concept supports a multifaceted approach, recognizing organ systems work together and influence one another. This also suggests that specific pathologies cannot be described by studying one aspect alone. As scientists, we have an obligation to get the most out of our animal models, and by adding additional endpoints, there's a good chance we can improve the quality of science, provide a holistic view of the disease model being studied, and actually participate in science that may result in a higher impact. And at Data Sciences International, collaboration that benefits research interests globally is our primary objective. In fact, we are proud to be part of the Harvard Bioscience family, where several companies, including PanLab and Colburn Instruments, are well equipped to assist with behavioral approaches. I encourage you to reach out to us to see how we can work together to help you achieve your research potential, get the data you need, and be confident in your approach. These references here are also available on our website at datasci.com. And with that, I thank you for your time, and we'll pass the controls back over to Liam for another audience poll.
All right, Mike, thanks so much for sharing your presentation with us today. And with that, we are just going to move on to the Q&A. Let's jump right in. So the first question here came from registration. And it's, well, in your experience, what are the biggest challenges for measuring or analyzing EEG? And Rob, maybe you can lead this one.
Sure. I think really the biggest challenge for our group getting started was figuring out how to really streamline the data analysis pipeline and pick bins to actually analyze everything and bin it into. Once we got the pipeline going, it was much more streamlined, but that was really the biggest hurdle.
Perfect. And Mike, you work with a lot of researchers, so I'm sure you have a good sense of common pitfalls and difficulties. What do you think seems to trip up scientists, and what kind of advice maybe can you offer to help?
Yeah, Liam, I think the biggest thing is that, you know, there always is an overwhelming feeling of how to handle large data sets. And the good news is that you really don't need to do this all on your own. Both of our technical support groups as well as our data services group are an excellent resource to help you better understand maybe a more efficient or streamlined study design to get the actual endpoints you want to see, or even work with them as a simple phone call from a consult all the way to a complete analysis and reporting opportunity. So it's don't do this alone. We recognize it's a lot of data, but that's why we're here to help you.
Perfect. And Rob, I know in your presentation you had a slide or two where you talked a bit about measuring activity in EMG alongside EEG. Do you have any tips or considerations that you can share?
As I mentioned, there's just so much data that you can pull out with these type study designs, which is really nice. You can look at circadian rhythm related changes with temperature and activity alone. You can really correlate that, with EEG or EMG. I mean, it's just a lot of flexibility. Again, a lot of data being generated that can be applied to, again, look at either therapeutic or adverse effects or disease progression in an animal model. The temperature and activity are really nice additions in that regard.
Perfect. And yeah, here's a question from Tavleen. She asked, what are the prospects of sleep linking to pain or pain-related disorders? Rob, maybe you can lead this one.
That's a great question, especially right now when we're thinking about the opioid epidemic and the transition from prescription pain medication to addiction. I'm not an expert in that regard, but I know there are a number of studies looking at both acute pain management and changes in sleep and chronic pain affecting sleep. So then you start getting into the multi-pronged questions. Is it the pain that's disrupting sleep or the pain management that might be affecting sleep? And of course, you could start parsing that out in animal models.
Perfect. Thanks, Rob. And so here's a question, maybe Mike, you could cover this one. So what other physiological endpoints can be measured using DSI technology and synced with EEG data?
Yeah, that's a good question. I mean, a lot of physiologic endpoints that are available can actually be combined as well, and that's often being requested more and more these days. But the beauty of that is, regardless if it's wireless or hardwired approaches, and I was glad to see in the poll here that there's an interest in both telemetry as well as tethered EEG. You can also add additional things like cardiovascular and respiratory related endpoints, including LVP, respiratory tidal volume and rate. But even beyond the actual physiologic endpoints that we can bring in, there's probably equal of importance relevant information coming from third-party pieces of equipment that can be monitored while doing your physiologic data collection. And these would include things like activity wheels, treadmills, optogenetic pulses, behavioral inputs. So it's really anything that has some sort of analog out feature capability we can bring in. And if you want to combine cardiovascular, CNS, and respiratory physiologic endpoints, it's very likely we can help you out with that as well.
Perfect. Thanks, Mike. And in the interest of time, we'll just make this next question the last one. Our last question today comes from Marwa. Here's a good question. So what do you think are the advantages of wired versus wireless EEG? Rob, maybe you can start.
Sure. I think it really depends on the study design and the question you're interested in. I think the advantage of wireless is that you aren't restricting the animal in any regard, so you're really getting a very normal, physiologically stable measure. With wired, you can certainly do shorter recordings. You can really keep them awake a little bit easier if you're looking, if you don't want that sleep component and you're just looking at arousal or some other event-related measures, and pairing it with other neurostimulation type devices. Tethered certainly is a good route. It really just depends on what the question is at hand. They both have their strengths and drawbacks.
Perfect. Thanks, Rob. And Mike, do you have anything to add on top of that?
Yeah, just real briefly. I mean, and Dr. Gould summed it up quite nicely. With a wireless, fully implantable, you reduce the stress. It's a nice approach. It ranges from small to large animals, and it provides chronic data collection. But there are individuals that stick to a tethered approach as well for a variety of reasons. Again, it's more of acute data collection, but it enables us to do more electrode pairs, maybe a higher sampling rate and a larger bandwidth. And you can incorporate that into a variety of different applications and other pieces of equipment. The summary in all of this is that regardless if you're doing a hardwired or wireless approach, the software platform and the analysis capabilities that Dr. Gould so eloquently spoke about can accommodate both. So as your research evolves, like I alluded to earlier in the presentation, as your research grows or changes or shifts focus, the likelihood is that we can still provide some guidance and help you walk through those processes from a hardwired to a telemetry approach.
Perfect. All right. Great answer. Well, thanks so much, Rob and Mike, for the really excellent presentations and discussion in the Q&A session.
My pleasure. Thank you.
Yes, thanks for inviting us.
And thank you to all of the attendees for joining us today. And of course, a very big thank you to our sponsor, DSI. So in closing, thank you again for taking part in this Inside Scientific webinar, and we look forward to having you with us again soon. Have a wonderful day, everyone.